Sorry. Hi everyone again. So we now welcome Ashley Parker from the Max Planck Institute for Biological Intelligence. So Ashley is going to talk about the factors guiding the emergence of shell-dwelling cichlid larvae from their nests. Sorry for my English. You can go, Ashley.
Hi, everyone. As they mentioned, I'm Ash Parker. I'm from the BioLab at the Max Planck Institute for Biological Intelligence. And I'm excited to introduce this very charismatic little fish and their very interesting parental care strategy to you today. I hope everything works with the presentation. It didn't fully present as we expected, but we'll see as we go along. So cichlids are a group of freshwater fish that predominantly hail from these three lakes in East Africa. And Lake Tanganyika is where the group originated from and has the largest diversity of genera. And along with this diversity in species and genera comes a great diversity in their behavioral repertoire. So we see that they're often very aggressive fish. They have hierarchies. We see different mating systems, polygamous, monogamy, harems. But one thing that is shared across all the cichlid species in the world is that they practice parental care. And how they do this is in one of two different strategies. So they can either be mouth brooders, where they collect their larvae or eggs, and then the larvae grow up inside the cavity of the mother or sometimes father's mouth. Or they can be substrate brooders, in which the female will lay her eggs on some sort of substrate, like a rock or in a crevice. The eggs are sticky, and she will then, along sometimes with the father, guard these eggs and offspring until they're old enough to fend for themselves. Now, there's a specialized form of substrate brooding called shell dwelling, which is the species we're interested in, is classified as this. And this is really a unique strategy within Lake Tanganyika. So essentially, on the floor of Lake Tanganyika, there's these beds of these abandoned snail shells. And this is so unique because Lake Tanganyika has a really high calcium carbonate concentration in the water. And so the shells don't actually dissolve as they normally would in another lake. And this provides an excellent opportunity for these tiny fish to make use of these kind of protective little environments in which to raise their offspring. And I work with one of these species, Lamprologus ocellatus, which is an obligate shell dweller. And that means that it won't lay its eggs in anything other than a snail shell. So there are 4 different phases of parental care in these species. The first is the female lays her eggs inside the shell. These then hatch and exist in the lower whorls of the shell with these big orange yolks that sustain them while they're not eating. And at some point, the larvae are seen to emerge from the shell for the first time, but then they will go back and forth into the shell for about a month. And at that stage, after a month, they actually move from the mother's shell across to the father's shell and then continue to be looked after by him. So for my PhD, I had 2 main questions. The first was I wanted to understand what the behavioral development of these larvae was inside the shell and the interactions with the mom. And then secondly, I was looking at this very specific behavior of when the babies or the larvae leave the shell for the very first time, and whether the mom is playing an instrumental role in that emergence time, or whether this is purely a developmental thing for the larvae. So in order to do this, we 3D printed these snail shells, which basically were cut at the back, which allowed us to then see what was going on inside, but the mother was still able to use the shell as she normally would. We then put a male and a female in a tank. They each get a shell as they would normally in the wild, and we cover the back of the shell with this black plastic, which is infrared penetrable, so we can use an infrared camera to see what's going on inside the shell. And then we film the female's shell for 11 days from when the eggs are laid, taking images every 7 minutes and also some videos in between. So here's a really fast video of, it's like a time lapse over the 11 days. It's going to go very quickly just to show you the basics. So the orange things are the larvae, and you see that once they hatch, they kind of spend the majority of the time in this deeper chamber of the shell, and they get more mobile as they get older. And then eventually they start moving up and using the whole shell, as well as exiting and coming in and out of the shell. That was very quick. Here are some snapshots of the different developmental stages. Here, DPF means days post-fertilization. But of course, we need a way, we can't just look at pictures, so we wanted a way to quantify this, and we used computer vision or specifically object detection, in order to track the individual or pick up where the individual larvae are in space and time for the duration of our study. This plot here is a very important plot, so I'm going to walk you through it because it's important for the whole talk. So essentially, we break down our shell into these three different chambers. We have the entrance chamber at the top right here, then the laying chamber, and the deep chamber. And then in the plot, we have a row for each of these chambers, and each vertical line represents a single time frame. And the intensity of color, or the alpha value, represents the normalized proportion of larvae in that chamber at that time. So what we see here is that initially they are eggs, and they are in this laying chamber. And at 3 days post-fertilization, they move down to this deep chamber, where, as we saw in the video, they kind of hang out there for the entire period until we see at day 9, they start moving up again into this laying chamber and into the entrance chamber. And this red line marks is an automatic mark for when we see that they leave the shell for the first time. And that's 9 days. When we repeat this over many, many, many different clutches, it is always 9 days, which was what we found so fascinating and why we wanted to look more into the mechanisms of what is controlling or regulating this 9-day emergence, the precision of this 9-day emergence. Of course, there's another player in this inside the shell dynamic, and that's the mother. So here we have a similar plot, again, the 3 different chambers, but now it just marks the presence or absence of the mother's. In this case, we recorded her head position. And I think the first thing you'll notice is that there are these clear like bouts where she's present for like continuous periods. And that corresponds to her nighttime occupancy. So basically, the mother spends 100% of her time in the entrance of the shell at night. Since we want to understand whether she's playing any sort of role in the emergence time, we wanted to see if there were any changes in her behavior across the duration of the parental care. And so instead, we look just at the daytime activity, which then varies as the larvae get older. So this is the age of the larvae at the bottom. And we see that the mother spends significantly more time in the shell in the first 3 days after fertilization. And then there's this drop off, but there doesn't seem to be anything significant happening at 9 days post-fertilization when the larvae would normally emerge. However, we weren't convinced that the mother wasn't playing a role, so we did an experiment which we termed the momectomy, where we essentially removed the mother and the father at a specific age, and then we continue to record what happens with the babies and the emergence time. And so what you see here, orange above the figure, represents the presence of the mother, and the X is when we remove her. And clearly, you can see that almost as soon as we remove the mother, the larvae are emerging from the shell. Again, we do this at 5 days post-fertilization, and again, the larvae emerge for the first time soon after the mother and father are removed. So she's clearly playing an important role, but we first wanted to see, we know that the mother performs these fanning-like behaviors where she basically flushes in fresh water into the shell at regular intervals. And we wanted to see whether if we continued to provide the larvae with fresh water, if this would then replace the function of the mother in the shell. So we essentially supplied fresh water into the deep chamber of the shell, And we found that when we did that, so that's fresh water being supplied is indicated by the blue line above the graph. And we've then removed the mother at 7 days. And we found that this then restored the emergence time of the larvae to 9 days post-fertilization. So this suggests that actually the only function the mother was playing was providing them with fresh water, and the larvae needed to leave because of the lack of oxygen or buildup of toxic chemicals in the shell. However, we weren't sure, maybe it could be that the mother stops fanning, for example, at 9 days and is still somehow indirectly playing a role. So what we decided, like in her behavior adjustments. So what we decided to do was to try confuse the mother. So we took 2 clutches, one of 6 days and one of 4 days, and we switched the clutches so that we ended up with a mother that had offspring that were 2 days younger than she would imagine her offspring to be, and one that had offspring 2 days older than what she would perceive. And these are the results. So first we're going to look at a mother that has babies that are 2 days younger. So essentially the expected age for the mother is on the bottom and the actual age of the offspring is on the top. And what we see here, the mother should expect the larvae to emerge at this point, but they don't. They emerge at their normal 9 days. So that suggests the mother's not changing her behavior around her perceived 9 days, or it could be that she's in fact able to deduce the needs of the offspring and she adjusts her behavior accordingly to that. However, the other situation told us otherwise, and we found that in this female, she's got larvae that are older than she expects. So their behaviors are now, in her perception, premature. So at her perceived 7 days, the larvae are actually 9 days, and we see that they don't emerge as expected. And instead, the emergence day gets pushed back a day to day 10. And I then was like, okay, well, what's happening? Why are these larvae not emerging? Is it because they don't want to? Or, this was an interesting finding. And here I have a great video showing exactly what was happening. So this is now that 9 days when they would normally emerge. And we see the mother coming in, picking up the larvae and really putting them back down into this deeper chamber of the shell. So she's, and that fanning with her fin is also creating a current that basically pushes the babies back down into these deeper chambers. So she clearly has an expectation and she has her own independent clock that she's following. And in her mind, these guys are prematurely trying to leave the shell and she's not happy with that. And this is just multiple different scenarios I've compiled where the mother's coming in constantly to try and get these babies to get back down into the shell. But this still doesn't answer what is the so now it seems to suggest the larvae at least have their own biological clock, and we wanted to figure out what is the mechanism behind this this biological clock. So what is changing in the offspring, because it's quite an abrupt change, and normally in development things are relatively slow, like developmental changes happen slowly, but this is a very abrupt, distinct change, and we wanted to see if we could figure out what this is. So the first thing we thought about was the fact that inside the shell it's a very dark environment, and this is quite a big change for the larvae to then move into an environment towards the light. And so we set up this, along with one of my colleagues, we set up this phototaxis assay. So essentially, we're allowing the offspring to choose between a light side of a box and a dark side of a box where they want to hang out. Again, we used this infrared penetrable plastic so we could see through the whole box. And here's a video of the larvae swimming around. So we can clearly see, and they have the choice to go in either side of the box, and we can then track the larvae over time. So we do this from day 7 until day 10, because we know that the emergence time is typically day 9, and we want to track their behavior. And what this figure shows is the percentage of larvae that are found in the dark. So we keep them in clutches of about 5 or 6. And here we find that the majority of the larvae are hanging out on day 7 in the dark, And this continues on day 8 with a slight decrease to more potentially spending time on the light side. But really on day 9, we see that there's actually a preference for light. This then goes back to a preference for dark at night, which this black bar below indicates. And then again on day 10, we see this preference for light. And if we compare pre-emergence daytime activity to post-emergence daytime activity, we see that there's a light preference happening, like a significant difference with light preference happening post-emergence compared to dark preference pre-emergence. So with that, I would like to summarize that we found that shell dwellers raise their offsprings in the confinement of these shells. The larvae remain in the snail shell for 9 days specifically until emergence, and that the babies will leave if they don't have a constant supply of fresh water that the mother typically provides. Also that the mother and the offspring have independent clocks set to 9 days for emergence, and that these two clocks need to be aligned if they want emergence to occur as it would naturally. And then emergence from the shell is correlated with a switch from light avoiding to light seeking behavior in these larvae. And with that, I would like to thank all my colleagues and the lab support, as well as some friends and professors that have had input.
Thanks, Ash, for this presentation. So while we are waiting for, we already have some questions. So a question from Amanda Facial. Would be interesting to know how many males were involved in the breeding. Were all mothers fertilized by the same male?
No, they weren't. So some experiments we did a normal experiment and a momectomy experiment with the same male and female couple. But then, so as like a direct comparison, but between the experiments, besides like those occasional experiments, then we use different males and different females.
Thanks. We also have another question. Do you think emergence could also be affected by circadian cycle? Do you think the mother would change her expectation on emergence if the light cycle changed?
Yeah, so we've definitely thought about that. I guess we want to know how the mother is keeping her independent clock, whether she's counting the nights or days, or whether it's perhaps a hormonal change that, egg laying, like releasing eggs, could cause this massive hormonal change, and then those hormonal changes gradually change over time, and once they're below a certain threshold, she no longer tries to keep the larvae back in the shell. So we know it's we think it's one of these two things. Changing the circadian rhythm is really, really tough because you have to, I guess you would have to keep the fish on that entrained time for a while before breeding. And since we house our fish in the same facility that we record from, because our recordings take so long, they have to be in the water has to be changing all the time. So we do all our filming and everything in the same system that we house in. It's a bit difficult because there are other people doing other experiments for me to change the entire circadian rhythm of that. We have spoken about it, and it's definitely something on our radar.
Thanks. We have another comment saying from AB George saying that great talk, very cool method on videos. So this is not a question, just a nice comment. I have a question for you. I think it's a naive question because I'm not working on fish. Do you think the mother would recognize the shell if you move it, or would she recognize the babies if you move the shell? Like, since she's taking care of the shell and the babies, can she recognize them if you mix with another mother shell or something like this?
So I think that they probably more likely use landmark cues to get to their shell. If you moved it 5 centimeters or something, I don't think it would be a big deal.
Okay.
But if you really moved it to another part of the tank, I I think when she's in a maternal mode, so one, we know she doesn't recognize her offspring, right? Because we've done that clutch switch experiment, and they accept the offspring. There's no problem there. I think if it was within a tank and we like had her shell in one position, we took it to the other side of the tank, and it was the only shell with babies in it, I think she would look after them because she's in like a maternal mode. If there's a whole bunch of other fish around, then that could get complicated because they're very territorial. They fight over their shells all the time, and she wouldn't feel that it was her shell. So I think it could get quite complicated. I think shell placement, like being in a completely different location, they would think it's not their shell. That would be my guess.
Okay. Thanks a lot. So I think it's time. It's the end of the question. You still have lots of questions. So I guess you can just check the question on Discord.